So our next talk is a proceedings paper, and the first author couldn't make it, and Jessica Li, who's which one? View. Okay. Slideshow. Okay. Jessica Li from UCLA, who's the senior author. She's gonna be presenting it. If you can move it on the other side of the screen, yeah. And then you. Oops. Okay. Yes. So here.
I can try it.
Do you know the shortcut?
Yeah, I know the shortcut.
Shortcut, the full screen.
Okay.
All right, Jessica.
Thank you very much for inviting us here and also for selecting our paper to be published by Bioinformatics. So today I'm going to introduce our recent work, which was developed by my PhD student, Vivian, who couldn't be here because of visa issues, unfortunately. And this paper is about a statistical simulator for single-cell RNA-seq data, and the purpose is to assist experimental design. So first of all, single-cell RNA-seq has been popularized over the past 10 years. So as we can see, from 2009 till now, there are more and more protocols and platforms that are being developed for sequencing individual cells. So as a biologist, many people wonder, how should I select the protocol or platform? Of course, many selection was done by convenience, but many times when you have a choice to choose between multiple platforms, the choice is critical. So as we can see, there are two major types of protocols. One is droplet-based and or UMI-based. The other is full-length. So they have their relative advantages and disadvantages. So for those full-length protocols, you get deeper sequencing depth per cell, but you sequence fewer cells. While for the 3' end counting or UMI-based protocols, you sequence many more cells, but you get very shallow profiling of each cell. So moreover, there is a trade-off. Given the same budget, there's a trade-off between the number of cells to sequence and the number of dropouts you obtain. So let's just use two platforms, Fluidigm C1 and Droplet Microfluidics, in contrast. So we can see one sequence hundreds of cells, but you get a million reads per cell, while the other sequence 10K cells, but you only get 100K reads per cell. So given the same budget, if you want to sequence more cells, you will get more dropouts. Regarding the experimental design for single-cell RNA-seq, existing methods mainly fall into two categories. The first category is to do some model-based or theoretical analysis to estimate the number of cells to sequence. So the simplest approach is actually developed by the Satajia Lab, who developed SURA software. So they have a web page for calculating or estimating number of cells to sequence by simply using a negative binomial model. And more recently, there is a paper from Barbara Engelhardt's lab at Princeton. This paper is still on bioRxiv, and they propose an estimator of the number of cells of each tissue, so to maximize cell type discovery. For those theoretical-based analysis, you need accurate knowledge about the cell type composition and cell type gene expression profiles. Unfortunately, for a lot of the new systems we explore, we do not have this knowledge. And also, for the estimators obtained from this theoretical analysis, it's really difficult to visualize how the design might work. So therefore, for biologists, they are not very intuitive. And the second category of methods use descriptive statistics. So this paper used the sensitivity and just vary the sequencing depth to see where it saturates. So the sensitivity is defined by the number of genes detected. So we see by this criteria, we will have saturation at about 1 million reads per cell. However, if we switch to a different statistics called the detection rate, which means that for each gene, at what number of molecules can it be detected? And we see here the saturation occurs at 4.5 millions per cell. So given two different descriptive statistics, we get different sequencing depth per cell. So we cannot easily unify these measures to guide our practices. And also, these calculations are not specific to biological conditions. So to address those issues, we propose this method called scDesign. And our idea is to do some realistic simulation so that people can use our synthetic data to test different experimental design settings and choose the setting that satisfies their downstream analysis goals. The input of our method can either be public single-cell RNA-seq data, which have been more and more widely available in public data sets, in public databases, or in some labs, they will generate pilot study data before they scale up their experiments. So then they can use their pilot data and as input into our method for designing the scaling up experiment. And this method will generate synthetic data that will hopefully mimic real data under the pre-specified experimental setting so as to assist experimental design and downstream method development. And we want it to be flexible in accommodating user-specific analysis needs. And the bigger advantage of using this instead of trying multiple pilot studies is that you can save experimental costs. So method. The key features of scDesign is that we want to leverage existing real single-cell RNA-seq data. And a key step in our model is that we use a gamma normal mixture model to account for dropouts. And this model was previously used in our imputation method called scImpute, which was published last year. And we found that from this plot of real data, we show that it can usually fit real data pretty well. And the fitness is actually better than the widely used non-negative binomial data, which directly models count data. In our case, what we did is that we did a log transformation, and then we fit this gamma normal mixture model. And there are two flexible modes for our method. One-state mode means that you just sequence a batch of cells together, while in the two-state mode, you sequence two batches of cells together, and they will compete for each other. So this is like the polling case. Okay, so I will first introduce the one-state mode, which is the basis of the two-state or multi-state model. So given an experimental setting, the budget is the constraint. So we fix the total sequencing depth, and we vary the number of cells. So if we can input one real single-cell data set from a one-cell state, then we will output a single-cell RNA-seq data set. So this is the diagram of our framework. So the first step is that we will estimate 5 parameters from a real data set. They include 3 gene-wise parameters and 2 cell-wise parameters. So for the gene-wise parameters, we have per gene variance, per gene mean, and per gene dropout rate. For the 2 cell-wise parameters, we have per cell dropout rate and per cell library size. And after estimating those 5 parameters, we will use sampling to simulate gene and cell-wise parameters for new cells. And these are in the dashed box. So with those simulated or sampled new parameters, we'll generate an ideal gene expression matrix in those new cells. And also, we will generate a dropout indicator matrix to indicate which values will likely be dropped out. So combining the ideal matrix with the dropout matrix, we will actually obtain a proportion matrix which indicates the proportion of each gene in each cell among the whole matrix. And then we will use multinomial distribution to convert the proportions into counts. So the output will be a synthetic gene expression matrix with entries at read counts. Okay, then we generalize this idea to two-state mode, so that in the two-state mode, we will have cell numbers, we will vary cell numbers of the 2 states, but we'll still fix the total sequencing depth. So the output will be 2 single-cell RNA-seq data sets, and the idea can be generalized to multiple cell states. So this is a simple summary, which basically means that we will use our previous one-mode approach to simulate the dropout matrix, which is after combining the ideal gene expression matrix with dropout indicators. So after we obtain those 2 matrices, we will combine them into a proportion matrix and then use multinomial distribution to simultaneously generate 2 count matrices. So the key idea is that in this multinomial distribution step, we are able to allow those two cell states to compete with each other for the total sequencing depth. Okay, so then to mathematically formulate the problem to make it more clearer, in the one-state mode, we input one matrix, and we have I genes and J0 cells, and the constraint is on the sequencing depth and cell number. Then when we simulate a new count matrix, we keep the same I genes, but now we simulate J new cells. In the two-state mode, we will input two real single-cell count matrices, and they have the same genes, but different cells. And we will constrain on the total sequencing depth and the cell numbers of each state. And then we will simulate a new count matrix for each state. So now I'm going to show the results of our method. So we studied 6 protocols that each have 3 cell types. So these are the 6 protocols. So you can see that they cover from Fluidigm-1, SmartSeq2 to DropSeq, 10x Genomics, and also includes SeqWell. And we consider 5 simulation methods. One is our method, scDesign, and there are 4 other methods in the literature. And they are not specific designed for experimental design, but they can also simulate single-cell RNA-seq data. So we did a comparison. So then how do we evaluate our method? So what we do is that for the real data, for each real count matrix, we randomly split the cells into 2 subsets. We use 1/2 to train each method. And then we use the other half to compare with the synthetic data by each method and see how similar they are. We compare the synthetic data with real data based on the distributions of 6 summary statistics per gene count mean, variance, coefficient of variation, 0 proportion, and per cell library size and 0 proportion. And so in total, we did 108 comparisons. And among those comparisons, our method is the most similar to the real data in 84 comparisons. So here are just some examples. As you can see, we use box plots to compare distributions. And the first box is real data, and the second box is our method. If you look at them side by side, you will see that we are the most similar to real data. We also compare the relationship between pairwise 2 summary statistics, and we are also very similar to real data, while some method like SPLAT is very off. So this is just showing the ranks of the different methods. So ranks being 1 means that it's the best performing method. And also, this is for SmartSeq2. For DropSeq, we also show very similar results. Although the distributions change a lot from SmartSeq2 to DropSeq, but we are still the most similar to real data. Okay, now let's look at the effect of experimental design. We consider 2 scenarios. In scenario 1, we consider cells from the 2 cell states are prepared as 2 separate libraries and sequenced independently. These correspond to many real scenarios. For example, cells collected at 2 different time points, or cells of the same tissue from patients and healthy subjects, so 2 batches, or cells of the same type but exposed to different experimental treatments. So in this case, we want to select the optimal cell numbers simultaneously for the 2 libraries to optimize the subsequent differential expression analysis. And here the constraints are the total sequencing depth of the 2 cell states. Okay, so given 2 input real matrices, and also a predetermined total sequencing depth, we will simulate 1 synthetic matrix for each state. We will perform DE analysis, and we will calculate target criterion set by the user. For example, it could be false discovery rate. Since this is simulation, we can know which genes are true DE genes. And here we define true DE genes as the top N genes ranked by this normalized difference. So here the parameters are estimated from real data, but in our simulation, we treat them as true parameters to define DE genes. And then we can have plots like this. So the first example is that we have the same 2 cell types to compare, and we look at the optimal cell numbers selected for different protocols. This is Fluidigm C1. So we see that given the set FDR, we vary the number of cells, and then we compare the precision, recall, and 1 minus false positive rate, the F1 type 1 F score and type B F score. We see how they change as the cell number changes. So then you can see, given the same FDR, we get one curve. And the interesting phenomenon here is that if we care most about precision, that is, we want our identified DNA genes to be very accurate, then you want to go for a small number of cells, because then you get very deep sequencing depth per cell. However, you suffer the recall. If you want to have the recall maximized, then there's a trade-off. If you have too few cells, the power is bad, so you cannot get a good recall. If you have too many cells to sequence, then the most information per cell is likely 0. The recall is also bad. So I think the recall is a realistic criterion for people to use, and somewhere in the middle is what you want. So here we see that if using the Fluidigm C1, the optimal number for recall is about 512. And if we move to InDrop, a different protocol, then the number of cells dramatically increased to 4,000, which also matches our expectation. Because for this protocol, we generally sequence more cells than in the Fluidigm C1. Okay, now we look at different cell types but the same protocol to see whether the design is reasonable. So SmartSeq2, 2 subtypes, which are not very different. So we see that here the recall gives us an optimal number 512. If we compare 2 cell types that are more different, then the number dropped to 256. That also makes sense. If we compare something more different, then we do not need so many cells. Okay, scenario 2. In here, we want to sequence 2 cells from 2 states together. So here, we only need to select 1 optimal cell number for the whole library, and the constraint is the total sequencing depth. So we still input 2 real count matrices, and we need a predetermined total sequencing depth for sure, and we will estimate the proportion of the 2 cell states. So in this case, we will still simulate 1 count matrix per cell. We'll do D analysis, and similarly, we'll evaluate the criteria. So this is the result. So first of all, we look at this Fluidigm C1, 2 cell types of these proportions. So these are the numbers we get. So let's look at recall. It's about 1,000. And if we move to a different protocol, InDrop, and interestingly, both 2 cell types are of smaller proportions in real data. So now you see the optimal cell number dramatically increased to this 16K number, so which is reasonable because here these 2 cells are not so enriched in real sample, and also this protocol need a large number of cells. Okay, so this is an interesting result to further validate our method. We want to validate the reproducibility of our method. What we did is that in this study, we used their data and we look at 2 cell types, but in different regions. So they're the same 2 cell types, but we look at the dorsal horn and look at the hypothalamus. And interestingly, the criterion we use to evaluate or to select the optimal number of cells is very consistent between the 2 regions, which suggests that our design is reproducible. And furthermore, we look at cross-study. So in those 2 studies, they both studied these 2 cell types, and they both used DropSeq. So if we use our method to do the experimental design, we see those curves are also very consistent, confirming the reproducibility. Okay, furthermore, we want to compare how computational methods work for differential expression analysis. So in this case, we consider 5 methods. The baseline is the 2-sample t-test. And we also consider MAST and scDE, both of which were specific designed for single-cell RNA-seq data. And DSeq2 and EdgeR were widely used for bulk RNA-seq, but later people added this ZingR to make it workable for single-cell RNA-seq. And we still look at those 6 experimental protocols. What we did is that we simulate a pair of rate count matrices with 5% DE genes to evaluate those computational approaches. And this is the precision recall curve for each experimental protocol. And in each panel, we have those computational methods plotted. Interestingly, you see that t-test does pretty well for SmartSeq2, Fluidigm C1, right? So t-test is not bad. But if we look at in-drop, drop-seq, 10x, and seq-well, then t-test is extremely bad. This is actually not surprising because for those 4 protocols, the Gaussian assumption, which is needed by the t-test, is clearly violated. While for those 4 protocols, the specific design single-cell methods have advantages. So to summarize this plot, we rank those methods by the area under the precision recall curve. So as I said, t-test does reasonably well for SmartSeq2 and Fluidigm C1. For the other 4 protocols, it's either edge-R, MAST, scDE. These 3 methods work pretty well, so which is also very consistent with our empirical observations. So because of time constraint, I will stop here for the result. But please note that our method can also be applied to compare other computational pipelines, for example, those for dimensionality reduction or visualization. OK, so this is the paper which was published recently. And this is the conclusion. So the method scDesign will simulate synthetic single-cell RNA-seq datasets that will capture gene expression characteristics in real data. And the experimental design will depend on users' analysis task, single-cell RNA-seq protocol, and cell population heterogeneity. We use cross-study comparisons to verify that single-cell scDesign leads to reproducible experimental designs. And our software is available on GitHub. And lastly, I want to thank my former PhD student because she just graduated, and she will join the Rutgers Biostats department as an assistant professor in this fall for her incredible work. And she has done also other works in my lab. She's a very productive student, and I also hope she will have a successful career. So I also want to thank our funding agencies and also the ISMB conference for inviting us. Thank you.
Unfortunately, due to time constraints, we are not going to be able to take questions for this one. Please find the speaker after the session. Our next speaker is going to be